I don't know, something happened last night when I uploaded the video. YouTube didn't like it, so I did it this morning. I think it's now up. And so, if you take a look, let's see where I put it. Way down here. And now, so it's there. Good. What might be helpful to you is, and I never thought I would ever say this, if you would subscribe to my channel, it'll inform you when things pop up there. And sometimes I will upload it, but I fail to get to the link on Moodle quickly. And so if you're subscribed, then you'll know when videos pop up, and they'll be there for you. And so you aren't dependent about having to go to Moodle to get to the videos. And so, yeah. My daughters love and watch YouTubers. I guess maybe, am I a YouTuber yet? I don't know. This is kind of odd for me. So, and I particularly don't even like listening to myself. It's kind of weird. So, but they're there. And again, they're there as a supplement for you as you study. Because I just say a lot of stuff. What's the important stuff? I don't know. It's all important, right? And so, but we'll have those available. All right, let's go ahead to the Lord in prayer, and we'll start in prayer. Dear Heavenly Father, I do thank you for this day. I thank you, Father, that it is the end of a week, and a weekend is coming. I pray, Father, that you will give us the strength, Father, to do all that is set before us today, that we will learn, that we will, Father, take time to do the things that we need to do. But God, in this weekend, I pray we find balance, that we find time to enjoy ourselves, time to rest, and time to spend with you. I thank you, Father, for your mercy. I thank you for your grace. I thank you, Father, for the provision that you provide to us. Let us be thankful of that each and every day. I ask this all in Jesus' name. Amen. Alrighty, so the cell is something you probably have learned. Oh, I don't know. When was the first time you actually talked about the cell? 3rd grade? 4th grade? And you made a, in my wife's elementary school, one of the grades makes a cell cake. And so every student has to bring in a cake with all of the organelles and everything. So the cell is not particularly anything that is exciting for you to learn about, but we just want to make sure that you do know all of the components. Now, here's the problem with the cell and the way we teach it from elementary school on. There is a picture. That is the idealized cell, and that does not actually represent what each cell looks like in the body. Every tissue type, if you look at a muscle cell, and you look at an adipose cell, they are actually very strikingly different. Now, they will have the same components, but in much different concentrations, and so you learn the idealized cell, that's in the textbook, but when you start to look at real cells, they're different. And so you need to know what is there, what can be there. As you go further, you'll start to differentiate. If you take histology, for instance, you really will start looking at how they look different, and the different combinations of how many organelles will indicate what kind of function's going on. And so if you're heading on to med school, you'll do histology at least in medical school, but if you take it here, it's kind of a helpful thing to do. But that's a misnomer in terms of how we teach the cell. It's all of this, but it is something that is the basic unit of life that we call, and it is capable of self-maintenance, reproduction, and all of the essential processes. Cellular components that are important are genetics, and so DNA, RNA. We'll spend a lot of time looking at this throughout the 3 semesters, and you've spent quite a bit of time in genetics looking at these components. Structural. Biochem is really going to be where we focus in on protein, and so we'll look at the structural components, how they're put together, how they're organized, how we look at them in different ways, and why the 3-dimensional structure is linked to function. So we'll spend a pretty good time, especially in this semester, on proteins. Lipids and complex carbohydrates can be structural in the cellular component, and so we will look at that, how the fats and the congregation of fats, and then how the sugars become linked in different ways are structural in nature. Some of them can be converted into energy sources, and so they're two distinct flavors of how we look at that. The cell, when we talk about it, oh my goodness, this is going to cause an epileptic shock. We have a lipid bilayer, okay? And so on one side you see the oxygens and hydrogens, the water, and on the other side you see the water, and in the middle you see the white and black are the carbons and hydrogens, and you can see that there is a true barrier. That the water doesn't like to, and very rarely does, invade where the hydrophobic nature is, and so there is a line of demarcation. And so we can control what is on one side and what is on the other. And in a lot of cases, the cell, talking about the cell membrane itself, and or the organelles, will control very directly what is on either side of that membrane. And there is a differential. This is essential to function. Okay, now here's the problem. If by chance we only had the hydrophobics, then the cell would be absolutely dead because we would not be able to take in nutrients, we wouldn't be able to give away wastes. So there is a high level of control of how we get things through asymmetrically through that membrane in a controlled manner. And so we'll talk about the lipid bilayer and how we use it to compartmentalize and then how to move things sequentially so that we can have function. We will do that by the use of proteins, and so we'll integrate our idea of proteins that allow passage of molecules. Very rarely are there any molecules that can just get through the lipid bilayer. What kind of molecule might be able to get through the lipid bilayer without having to be transported through? Water, even though it's polar, actually can leak through the membrane, okay, at a small rate, and that's important for the shape and function. So a little bit of water, although we do have, and most of the water is facilitated through a protein that comes in and out. What else might get through the membrane by itself without a transporter? A lipid itself, okay? Now, the kinds of lipids that we might get through or think about are natural hormones. Hormones that are very hydrophobic, mainly based upon cholesterol derivatives, can actually move through that membrane itself, and or drugs. A very hydrophobic drug has the ability to get through that membrane. But this is a limited set of things. Most things have to have a transporter of some sort. And so we will talk about how signals or molecules can get through that lipid membrane and how that is accomplished. All right? And so we will allow passage of actual physical things and or signals from the inside out to change what happens and what's going on inside that cell. Sometimes we can't make it through the membrane by itself through a transporter, so sometimes we ingest or merge larger items inside the cell through endocytosis and phagocytosis. And so we have mechanisms to get even larger things inside the cell, depending upon what needs to happen. Now, we talk about the cell membrane, we talk about the organelles inside, but what they are floating in is the cytosol. Cytosol is very important. It is the liquid or water-containing part. It contains a lot of soluble things, mainly the items that are, we think about proteins, salts, minerals, small molecules, and the biochemistry, a lot of it that we describe actually happens in the cytosol. And so that's an important part of the cellular component. Cells are classified as either eukaryotic, prokaryotic. This is probably something that you have heard about and have studied before. Just very simply, eukaryotic cells has a nucleus. Usually in complex multicellular organisms, the prokaryotic cells have no nucleus and usually are classified in the bacterial range. What I need you to know is if I gave you a cell, you could label it. Okay, so nothing more than that third grade class. I'm going to ask of you to be able to do that. But on this, the animal cell in the middle is what we are going to spend most of our time. Sometimes we compare and contrast to the plant cells, and we'll talk about that, especially when we compare oxidative phosphorylation and photosynthesis, both creating ATP. And then there's a bacterial cell, but you should be able to identify and label each one of those. I'm going to point out a few things. The ER. One thing that we know with the ER, there's rough and there's smooth. One thing I do want you to know about the ER is that it is a place where calcium is stored. This is going to be important when we talk about cellular signals. And so one thing you should associate to the ER is the sequestering of calcium. You may have talked about the cytoskeleton, but what we're finding is that the idealized picture of the cell also creates this idea that, okay, here's the cell, it's a big bag, and you got all this water and everything's floating around, and it's all random. And what we're finding is that the cell is much less random, and that there is organization of everything together to create bundles of functional proteins or units. And so if we talk about a pathway that occurs, usually all of those proteins are associated, bound together, organized. And it allows us to increase the speed and specificity of that entire pathway, whereas before we thought about it as the molecule substrate finds one enzyme, gets transformed, that product goes into another enzyme somewhere, and they're floating around and just kind of diffusing from one place to the other. But it's a lot more organized. Well, if you're going to organize organelles, then you've got to move them to certain places. And what allows us to do that is the cytoskeleton. It is a cellular structure. If we think about the cytoskeleton, it is similar to the metal girders inside a skyscraper. That is what gives rigidity, and it forms a pathway like a roadway in which we can move things. There are monomeric single proteins that under certain physiological conditions are by themselves. But under other physiological conditions, they bundle in long tubes that create essentially roads. And so we get microtubules and microfilaments from actin and from tubulin, individual subunits. If you actually look and we highlight or label the cytoskeleton, there's a cell. And do you see where all the green is? That's where the cytoskeleton is. It is pervasive inside that cytosol portion. Most of the time we don't ever see it because most of the time we don't highlight it. But it is very organized. Now, that organization can break apart and form very, very quickly. And so it allows a cell to have movement, but when it needs it, it allows it to have a very rigid nature. And so once we have that very rigid nature, those roads, then we can use proteins to move components around inside the cell. We'll use proteins called kinesin and dynein, which we'll look at in detail in a later chapter, but we will move vesicles and organelles from either the inside of the cell out or from outside the cell in, and we have certain proteins that move them in either direction to organize what's going on. It's an ATP-driven process to move physically the organelles around on those microtubules. And so we'll go into detail. We'll show how how the conversion of the energy causes a change in conformation of the protein and actually move. But the cytoskeletons are very important in organizing what's inside the cell and where things are. Now, most of the biochemistry that you will learn at this moment in time comes from this strategy. You've got a cell, and you've got a lot of things inside that cell. Okay? But you want to know how one thing inside that cell works. So this has been the strategy for biochemists all the way up to this moment in time, is to, okay, take a cell, and then take out that one thing you want to study so that you can put that into a test tube, control what that protein sees, and see what happens. And so you try to control as many of the variables as possible, change one thing, and then that change will cause a change in function, which then you can build a relationship. And that's what we do. And so we will isolate the smallest amount that causes a certain function, and we will reconstitute that in a test tube and do lots and lots of tests on it. Okay? What do you think are some advantages about that method, and what do you think are some disadvantages about that method? Because if I change one component in a full cell and I look at what happens, okay, things might happen. So if I change the pH, that cell may totally change what it does, right? And the outcome. Or I may take out one protein and change the pH and something might happen. Which one is going to give me more valuable information or easily obtainable information, do you think? The test tube or the cell?
I feel like it would be the test tube because you actually can go down to the specific area.
You can say this, that, yeah, I've changed this condition, it changes this protein because I see this change. I mean, that's direct, right? That's an advantage. Whereas if you change something in a whole cell and you see an outcome, where was that outcome influenced, right? It's direct. But with that direct nature, is there a disadvantage?
Yes.
What would be the disadvantage?
You don't know how it affects the rest of the cell.
You don't know how it affects the rest of the cell. Also, you don't know if something in the cell would have interacted in a different way that would have prevented that change you made to this one thing you identified to actually really occur. And so reconstituting something inside a cell allows us to control a lot of variables. That's what we want to do as scientists. But sometimes it can take you down the wrong pathway. Okay? And most of the time it doesn't take you down the wrong pathway, but sometimes it does. This can lead to two different laboratories studying the same thing to get two different results, depending upon how they process what they do. And so you start with a lot of times tissue. You isolate the cells you want, and so from that you break them apart. You do a lot of purification, centrifugation, lots of other types of purification that we're going to talk about, and you fraction them out and you isolate what you want to study. This is basically how the majority of that textbook that you have has gained the knowledge. All right? I did studies myself, and we were working with a protein called chemogulin, and there were two tissue types in which the chemogulin was very high in concentration based upon the weight. And so the two items, we got them from bull, were the bull's brain, which it was about yay big. I actually went to the slaughterhouse, got the brain, took it back to the lab, took out the bullet, and isolated my protein. Okay, so that's the bull brain. Or the other tissue type was a bull testy. One bull testy is about this big. Tells you a little bit about a bull. Okay, and so both of those started with the tissue type. They had high concentrations. First step in my process, cut it up, put it in a blender. Okay, and we would start the process to isolate the protein from that. Now, when I taught Biochem Lab, we did the same thing. Now, this is way before, the lab safety that we're like really, it's really important, because when you go into lab, what must you have on? Goggles and long pants and closed-toed shoes, right? All right. way back when we were a little bit less concerned about that. I would start them off. They would cut up the tissue. They would put it in the blender. One time, one student, she was doing the experiment but didn't screw on the blender thing really well, and she was wearing flip-flops at the time, and so she picked it up and it leaked. After that, she wore closed-toe shoes. So that was a good learning experience for her. But these are the kinds of processes that we do and lead to how we study these things. All right. In general, what I do need you to know, we have prerequisites for this course. I assume you've taken GChem. I've assumed you've taken at least Organic 1, and I've assumed you've seen genetics. So the things that you really need to understand and know, and I'll test you on in the first exam, are things about Lewis structures. I assume you understand that because the book is full of Lewis structures. You should know how carbon, hydrogen, nitrogen, oxygen bind, single, double, triple bonds, what those are. You should know your organic functional groups. I would go through the first chapter and look at the functional groups that are there. You should understand about chirality, RNS, cis trans, enantiomers, specificity, and know about electronegativity and bond strengths. These are just big conceptual ideas. You should understand that a polar bond happens because there's a difference in electronegativity, and in general the trend where fluorine is the most electronegative, and that kind of thing. And so these are just the kinds of talk and things that we'll talk about, the concepts. So please, if you read the first and second chapter, it goes over all of this material at the level that you need to know. But I assume that you've covered this pretty well in the courses that you've taken up to this point in time. Now, what I do want to introduce to you are the biochemical transformations, the kind of chemistry that happens. And so we can actually take all of the organic chemistry that we've learned up to this point and kind of distill them into categories that happen inside the body. And so we look at redox reactions. Okay? This is probably the most fundamental in terms of metabolism, the extraction of electrons. Okay? And so we're going to eat very hydrophobic, reduced material. Okay? You go down to Dunkin' Donuts, you start there, and you will end up with a very highly oxidized carbon, CO2, as you breathe out. Through that process, I'm going to take lots and lots of electrons. So redox is what we're going to look at. We will create and cleave bonds, mainly carbon-carbon bonds. We'll have internal rearrangements. That's a lot of what happens, where we may not even add anything or take away anything, but they rearrange. We are going to transfer things from one group to another, and we're going to condense things to make long polymers. These are the kinds of reactions that we'll see. Here's an example of a redox reaction, lactate to pyruvate. In essence, what we're going to have is a loss of a hydride and a proton. That's essentially what's going to happen. Or the gain of a hydride and a proton. Now, these classifications in terms of the kinds of reactions, the kinds of enzyme will really kind of identify what we're doing in this case. A dehydrogenase is the kind of enzyme that does these. Now, so what would the mechanism kind of look like? All right, I would actually look like this. Take out that hydrogen. I'm going to make this articulate. Some sort of base inside the enzyme is going to take this proton. This electron pair will form the carbonyl, and a hydride leaves. Now, if you write a hydride leaving in an organic class, that can happen. It is very unfavorable energetically. A negative hydrogen leaving is not a good leaving group, not one of your best. But inside the enzyme, there's going to be an electron acceptor, which makes the transition much easier to occur. And so we'll talk about the electron acceptors. On the other end, if I'm going to reduce, go pyruvate to lactate, I'm going to have a hydride donator, it will attack at the carbonyl. The negative charge will attack at the partially positive carbonyl, the carbon. Why is the carbon partially positive?
Because the oxygen is electronegative.
Oh, because the oxygen is electronegative. See, electronegativity comes into play. Okay? And so at that point in time, if I just drew a line that attacks the carbon, how many bonds do I have to carbon? At the end of my arrow movement? 5. Will you ever get anything right having 5 bonds to carbon? No. If it was CHEM-321, it'd give you a really big red box, okay? Or an X on the exam. And so I'm going to draw that it picks up a proton, but to articulate this to you, the first step would be the negative charge going on the oxygen. A negative charge on an oxygen, is that bad? No. It is high in energy, okay? But the oxygen is electronegative, so it is not that high in energy. Now, if I put a negative charge on a carbon, carbon is not electronegative. That would be very high in energy. And so having a negative charge on an oxygen temporarily, not so bad, even though it is the high energy state. And then the second would be to pick up the proton. But again, I'm a chemist, and chemists always try to do it as lazy as possible. So I just write the one arrow rather than the two. But I want to understand that that arrow movement represented 2, where the negative charge was temporarily on the oxygen before it picked up the proton. So this is a redox. Almost always it is the addition or the accepting of a hydride. And then a proton is always available to you because we're in a water environment. Also inside an enzyme, a base is always available because the enzyme will place an amino acid side chain that will work as a base. And I will generally write B with a pair of electrons to indicate that. Oh, look at this. I wrote out that mechanism all for you. Okay. And so usually a hydride is donated or accepted. Now the oxidation states of carbon can be multiple. And so a fully reduced carbon can be oxidized multiple times, or multiple numbers of electrons can be taken from that until we get to carbon dioxide, which is a waste product because no more electrons can be extracted. It is oxidized to its fullest extent. It's a waste product to us. But within the ecosystem, is CO2 always a waste product? No. What takes a CO2 and uses it? Plants. And plants will then create the sugars of which we eat to make CO2, and we've got the circle of life. Isn't that so grand, just how God created us to work in harmony? Okay? And so we move those electrons back and forth. All right. We can form bonds, okay? And so I'm going to form a bond between Z, the nucleophile, with W being the leaving group, okay? Now that Z could be a carbon, that Z could be multiple things, like nitrogen, but I'm going to create a bond, all right? And so I am going to, what kind of reaction is this? Would you classify this? I'm doing what? One thing for another? A substitution. That is great. What kind of substitutions do you know about? Version what? 1 and version 2. An S in 1 and an S in 2, right? What did I represent on this mechanism? S in 1 or S in 2? Which one went first? Now, you would assume A goes first, but does A always go first? No, we can be tricky. Okay, B could have gone first, and if B went first, it's not S in 2. Okay, and so I didn't indicate which one went first, which one went second. Okay, if by chance the first arrow is 1 and the second arrow is 2, all happening at the same time, that is an SN2 mechanism. Now, if you see below what's happening in that SN2 mechanism, the intermediate was a 5 or a pentocovalent intermediate, right? Temporarily, partial bond, 5 of them. And then the leaving group will leave what happens potentially to the stereochemistry for an SN2 attack? Inversion. Okay. Do I show inversion in the little animation? Do I show inversion in this? It looks like an inversion, right? But is it truly an inversion? No, because why? There's no stereochemistry. Okay, it's not chiral, so I can't see if it is. Okay, but one thing when you do look at a reaction, you're going to see the reactant and the product. Okay, when you look at the reactant and the product, and there is inversion of stereochemistry, that gives you an idea that that mechanism must have been an SN2 mechanism, substituted and inverted. If it doesn't invert at all, okay, and there is stereochemistry, then it can't go by this mechanism. Right? And so being able to look at the beginning and the end can actually give you an idea of how the mechanism might have occurred. All right, this is an SN1 mechanism dictated by a carbocation invoked in part of the mechanism. So, by chance, the leaving group just leaves, right? Left with a carbocation. Oh, let's go back to CHEM-1. What is the hybridization of this carbon? sp2. So what is its shape, molecular shape? Trigonal planar. So when you have a trigonal planar, the attacking nucleophile can attack on which side? Both. Statistically, how often do you hit each side? Equal, 50-50. So if you hit one side and the other and it becomes, the product is chiral, what kind of product do you get? Do you get a single product? You get two, and it's a mixture called a racemic mixture. Okay? And so in an organic flask, it's exactly what you learned, that it is a racemic mixture. Because that flat, there's no necessary indicator why one would be one side would be better than the other, and you get half and half. Inside an enzyme, though, if we have an SN1 mechanism, you get exclusively only one stereoisomer. Okay? It is because the catalyst, and you've learned this, or you may learn this in organic chemistry, can influence stereochemistry. And so what happens is, inside the enzyme, I bind the substrate. I bind the reactant. Okay? It may get flat, but it's still bound. So one side is blocked by the protein, and one side is the only side that reactions can occur. And it optimizes the reaction so that it hits from only one side. And so we have one stereoisomer. Almost exclusively in organic reactions, what you see is retention of stereochemistry. So you have a substitution, it is R at the beginning, and something substitute, and it's R at the end. The only way that could have done that type of reaction would have been an SN1 mechanism, because if you did the SN2, you would have converted it to an S. Internal rearrangements. Excuse me, glucose 6-phosphate, fructose 6-phosphate. If you count carbons, hydrogens, nitrogen, if you count nitrogens, 0, and phosphorus is in this, you find nothing has changed in terms of the numerical number. But you've changed something dramatically from an aldehyde into a ketone. Okay? That happens a lot. The kind of enzyme that does this is an isomerase. The mechanism for that is to deprotonate and protonate until you get to the final product. Take away the proton, add a proton. Take away a proton, add a proton. Take away a proton. And you have to do that in equal number And a lot of reactions occur this way. We have to change the isomer and the isomerization to do that. And it's just simply deprotonate and protonate. And so that's one class. Another class that looks complicated, but it really isn't, is a transfer group. Okay? A lot of times we're going to use the term activation in this. And what we're going to do is we're going to take one group and transfer it from a carrier to an accepting group. Okay? So let me give you an example. All right, the carrier molecule is in this case an ADP. Okay, what it's carrying is a phosphate. So when you put the carrier molecule ADP and a phosphate together, it's ATP, triphosphate. Okay, because the one thing that's going to move is a phosphate from one entity to the next. Okay, so it's carrying around a phosphate that can be transferred. In this case, glucose is what's going to accept that phosphate. And so the phosphate is going to transfer from the ADP and put onto the glucose. All right? The ADP is just the carrier molecule. The phosphate is what's being transferred, and the glucose is the accepting molecule. We're going to do a lot of these things. We're going to transfer things a lot of times. The artist is wonderful, but cannot write a mechanism to save their life. Okay? All right. In the product, there's no hydrogen, but they don't show the hydrogen being taken away. I'm going to take it away. This electron pair becomes the electron pair that attacks. Every time I attack something that has a carbonyl, the intermediate has to be first. The electron kicks up. In this case, it will form a pentacovalent intermediate. The electrons will kick back down, and the leaving group will be that pair of electrons onto that oxygen to create that negative charge there. And the oxygen that is right here is that one. Oh, that's color-coded. And then the three oxygens, 1, 2, 3, become the 1, 2, 3 in that particular case. ATP is going to be used a lot of times because that phosphate will represent a wonderful leaving group for another reaction. Phosphates are wonderful leaving groups. And by having that on, it will allow an attack somewhere and the phosphate to fall off. This is how Hess's law is actually applied. Remember we said if ATP is broken down, it gives oxygen energy, and I can add it on. Well, I don't just break it down and I don't just add it on. What I do is I allow the ATP to transfer the phosphate temporarily onto the entity that I wanted to have happen for the reaction to occur. And so it becomes part of the reaction. The two reactants react. Ultimately, the Hess's law will be what the resulting sum of the two reactions are, but they are processed sequentially in enzymes. Last, condensation or biopolymers. We will make proteins. We will look at sugars being polymerized, and we'll look at a lot of these types of things. Mainly, the types of polymerization is this. I take out water or I add water. And so if I take out water, I form the bond. Up at the top would be an organic reaction that you might do in an organic flask, in a round bottom flask, where a water could be taken away. Now, what that would look like is this. A base will take that proton, these pair of electrons will attack. Electrons kick up, electrons kick down. Then this hydroxide, when it falls off, picks up a proton. Essentially, water comes off. Now, that occurs, and that can occur, and you can put those in an organic flask and you boil the mess out of it, reflux it for hours and hours and hours, and that reaction will occur. The activation energy is very, very large. Okay? In the body, what we're going to do is put it on a transfer protein, This will be the unit that will be transferred, and it makes the reaction facilitate a lot easier at a temperature, at a much less temperature, much less extreme pH. And so this is a combination of condensation with a carrier protein or carrier group to transfer something. In this case, tRNA to transfer RNA. But we will form the same intermediate with the same outcome at just being facilitated by a carrier group. Okay. And so we will look at lots and lots of different types of reactions, but almost every type of reaction that you look at from this point on in terms of all mechanisms will fall into one of those 5 categories. Very simply. And in essence, what we're going to do is we're going to see the body does the same reaction with lots of different molecules. It's just basically the R groups are different. And so if you look at a reaction, even though the molecule looks this huge on a piece of paper, and it transforms into this thing over here, if you look at just where the reaction occurs, it looks the same for lots and lots and lots of different reactions. Don't get scared of the R's. Okay? In organic, what they did for you was when they taught you a reaction, they put a carbon and they put either 2 lines and didn't put anything else, or they actually put R's, and they said, it doesn't matter what's on the edges, this is what's happening in the reaction. What freaks people out in biochemistry is they see all of this other stuff and they think it's harder because it's bigger. No, the reaction's still only happening around 1 bond. So don't let the largeness make you nervous. Focus in on what's actually happening. Look at the beginning, look at the end, see what moved. You'll see most of it didn't move. Don't care about that. You want to see why the bonds change where they did. And that's a very small portion of that molecule. All right. Individual units are wonderful and great, but in the cellular component, most of what we look at are what are called macromolecules, or larger molecules. So proteins, even though they're made up of individual amino acids, which weigh about 110 grams per mole, the actual protein that is functioning are between 5,000 and 1 million grams per mole. They're much larger, and we need that largeness for the function. It's just a polymer of amino acids. The nucleic acids get much, much larger. Their molecular weights can be up towards of 1 billion grams to get 1 mole. Now, you don't have a mole of DNA in your body. That would be 1 billion grams. That would make you a little heavy. Okay, so little goes a long way. All right. The other macromolecules are polysaccharides, long chains of sugars put together. We love single and disaccharides. Those are kinds we eat. Those are kinds are sweet. But when they are stored or structural, they get into millions of molecular weight. And so we get polymers of sugars. And the last thing is weird, lipids. The polysaccharides, the DNA, RNA, the nucleotides, the proteins are all covalently linked and are rigid and very large. The lipids are considered a macromolecule, but look at their molecular weights. They're small. They are individual molecules that form aggregates that work as a big, large functioning group, but they do it non-covalently. What holds them together is the water polarness pushing them together to act that way. They do have hydrophobic interactions among them. So they are functionally a large working group, even though they are distinct in that they're not covalently linked. And we will see the different polymers made up of smaller units that are individualized. Phosphate seems to jump the gamut on all of these and are part of all the macromolecules. Now I talk about structural hierarchy here because we're going to study biochemistry in different ways. Sometimes we're going to look at the individual molecule and know something about that. But then sometimes we're going to talk about how they interact amongst themselves and a little bit higher structure. And then sometimes we're going to say, okay, make big groups, and then sometimes we'll talk about the whole cell itself. And so I will teach different things at different components, and we will keep usually between level 1 and level 3 a lot of times for the working, but eventually everything works towards level 4, and so. Do you have to actually identify where things are and which level? No, but I do want you to take a look. We look sometimes like a microscope. Sometimes we look at something very, very intensely, and we have to know that. But then we have to kind of get a bigger picture. How does that work in context? And then sometimes we have to get a bigger picture. And a lot of times, a lot of people in evaluations, just in general for classes, sometimes I didn't get the big picture. Yeah, you really, this is important. Sometimes we do focus in on detail, but if you know detail without context, that's not a good way to go about it. And so we always have to learn our detail in terms of a larger scope of why that is important and how it all works together. I attempt to do that at times. Sometimes I'm really, really successful, sometimes not, but I will always go back over it in office hours. Okay? But we do want to make sure that we can think about this in a higher level about why this is important, even sometimes when we're in the weeds. We will spend a little bit of time now, but a lot of time later in information transfer and talking about how that those work together. And we've come up with terminology that is relatively new in adoption because so much data is out there now that they form whole areas of study. And so for a long time, the genome was the holy grail, okay? We wanted to understand because we knew the DNA instructed how everything works, so we were looking for the genome, and we got that, all right? One thing about the genomic work, it was one of a very small handful of governmental projects that were done under budget and under time. Very few governmental projects look at any road being built, okay, ever is it under budget and under time. One of the other ones that are pointed out is the Hoover Dam. The Hoover Dam was built under budget and before the timeline. Everything else, a lot more budget. All right. Then we now know the DNA. Now we're looking at RNA transcripts and the transcriptome. We're finding a lot of information. And then proteins, the proteome, these are areas that are more active in research at this moment in time. Yeah, we know all the DNA, but we thought that would solve everything. No, it didn't. And so we're starting to look at complexities that arise from that. And so these are our active areas of study. All right.